I'm gonna talk about today the mechanism of statin to prevent hepatocellular carcinoma. So today, for the background, I will briefly introduce what is hepatocellular carcinoma is and why it is important. And also, I will introduce several studies showing that lipophilic statins reduce HCC risk in patients with liver disease. And then I will explain what is prognostic liver signature, because I used this methodology for my study. So the aim of my study was that investigating the differential actions of lipophilic and hydrophilic statins in the chemo prevention of hepatocellular carcinoma. And I will show you my study result. And next, I will briefly introduce what is my next step. So I will develop a herbal medicine preventing non-alcoholic fatty liver disease-related HCC. So hepatocellular carcinoma, HCC, ranks 6th in incidence in Korea. And cancer is the #1 cause of total death. And HCC is second cause in cancer-related death. And the prognosis of hepatocellular carcinoma is not that good because the 5-year survival rate is under 40%, and 10-year survival rate is just under 20%. And also, the socioeconomic burden of HCC is high because of higher mortality rate in productive population around 40 to 50 years old population. And this is the meta-analysis showing that lipophilic statins reduce HCC risk. In this study, the authors showed that statin users were less likely to develop HCC than non-users. And also, when we divide the statins into lipophilic statin and hydrophilic statins, lipophilic statins only shows statistical significance compared to hydrophilic statins. And also, when it comes to the individual statin, only atorvastatin showed statistical significance in decreasing HCC risk. And another statin like simvastatin and rosuvastatin, pravastatin, and fluvastatin didn't show the statistical significance in decreasing HCC risk. So this figure is the simple example of that lipophilic statins reduce HCC risk. So as you can see here, this lipophilic statin use, this dot line shows lower incidence of HCC compared to non-users. But in contrast, hydrophilic statin users didn't show significant difference between the control group. So this figure very simply shows that lipophilic statin use is associated with lower HCC incidence. And I will introduce what is prognostic liver signature. I will call PLS. Prognostic liver signature is a gene expression pattern in the liver tissue of the patient with liver disease. So when patients have HCC high risk, the upregulation of HCC high risk genes and downregulation of HCC low risk genes, the patient will be predicted to have high HCC risk. In contrast, when patients have low downregulation of HCC high risk genes and upregulation of HCC low risk genes, they will be predicted to have lower HCC risk. So as you can see here, the orange line means the high HCC risk population. So this population shows higher probability of HCC, but they show lower probability of survival. But the patient with low HCC risk, they show lower probability of HCC and higher survival rate. So this recently there is a prognostic liver secretome, the so-called PL-seq. So in this system, the blood markers like VCAM, IGF, BP7 is measured, and then the expression, the concentration level of these blood markers can be used for predicting HCC risk, similar to prognostic liver signature using gene expression pattern in the liver tissue. So this is the technique that can be used for non-invasive diagnosis for cancer, the patient with liver disease. So in my study, I used this PLS system in cell culture system to study the efficacy of many drugs inhibiting carcinogenesis. So as you can see here, when the hepatitis virus and ethanol, which mimic alcoholic liver disease, and free fatty acid treatment, which mimic non-alcoholic fatty liver disease, these treatments increase the PLS score. So you can see the red color is increased HCC risk. So it is red color means high HCC risk, and so orange color means high HCC risk, and green color means low HCC risk. So these virus treatment and ethanol treatment and free fatty acid treatment increase the HCC risk, which means high PLS score, but many drug treatments reduces the PLS score. We can see with the green color or pale color. So using this PLS system in cell culture model, we can screen which drug is best for the chemo prevention in cell culture model. So now the aim of my study was that investigating the differential actions of lipophilic and hydrophilic statins in the chemo prevention of HCV, induced HCC. I used HCV infection model to induce the PLS score, to induce high HCC risk. So I infected hepatitis C virus to hepatocyte-like cells, and then I treated 4 statins. The 2 statins were hydrophilic statin, and 2 statins were lipophilic statins. And then I measured the PLS score. So as you can expect, the HCV treatment significantly increased the PLS score, which means high HCC risk. But the statin treatments decreased the PLS score, but especially the atorvastatin very significantly decreased the PLS score compared to other statins. And I was interested in the differentially expressed genes modulated by hydrophilic and lipophilic statins. So there were around 200 to 300 genes modulated by hydrophilic and lipophilic statins, and around 100 genes were modulated by both hydrophilic and lipophilic statins. So these 104 genes were mainly involved in cholesterol metabolism, because these hydrophilic and lipophilic statins just inhibiting cholesterol synthesis. And I looked into these 66 genes. These genes were the genes modulated by atorvastatin only. So these lipophilic specific TEGs were like this. And also, again, I can found the atorvastatin significantly modulate these genes. And also, I found that these genes were mainly associated with YAP pathway. So, so this YAP pathway, when this YAP pathway activated, the fibrogenesis and carcinogenesis in the liver is induced. So these statins inhibit HMJ-CoA reductase in mevalonate pathway. So the statin treatment reduced the level of mevalonate and another intermediate product of in mevalonate pathway, GGPP. So the decreased level of this mevalonate pathway product induces phosphorylation of YAP. It results in the degradation of YAP protein and also the cytoplasmic sequestration of YAP. So this phosphorylation of YAP, it means deactivation of YAP. So when I treated atorvastatin to the cell, the phosphorylation of YAP is increased, which means deactivation of YAP. And also when I treated atorvastatin and GGPP, or the one of the intermediate in mevalonate pathway. So when I treated atorvastatin, the expression of CTGF and CYR61 is decreased. The CTGF and CYR61 is the target gene of the major target gene of YAP pathway. We can see it here. And when I supplemented GGPP, we can see the reverse of the inhibitory effect of atorvastatin. So this result shows that the statin inhibits YAP activation through a mevalonate pathway, specifically inhibiting HMG-CoA reductase. And also because the atorvastatin significantly decreased PLS score compared to other statin, I was interested in these genes regulated by atorvastatin only. So I came up with that there should be another mechanism of atorvastatin other than HMJ-CoA reductase inhibition. So I searched many papers regarding the other targets of statins other than HMJ-CoA reductase. So I found that there are many other targets of statins. For example, pravastatin and atorvastatin is the inhibitor of histone deacetylase. And I focused on this RXR alpha because atorvastatin only acts as the stabilizer of RXR alpha. The reason why I focused on RXR alpha, another reason was that the RXR alpha, a truncated form of RXR alpha, promotes a PI3K kinase and AKT pathway, and also promote the NF-kappa B mediated inflammation, and also inhibits apoptosis. So this complex mechanism of TRXR alpha promotes carcinogenesis. So targeting this truncated RXR alpha has known for promising cancer therapy strategy. So I look into the AKT phosphorylation to see the activation of AKT. So when I treated atorvastatin and other statins, in case of atorvastatin, the phosphorylation of AKT is very significantly decreased, but other statins didn't show that significance. And also, when I removed RXR alpha in the cell using siRNA, the statin treatment didn't show significant decrease of AKT phosphorylation. So these results show that the atorvastatin suppresses AKT phosphorylation, AKT activation in RXR alpha dependent manner. So, and additionally, I was interested in these upregulate unregulated genes by atorvastatin, and I found several MT2A and MT1A, these blah blah these genes. So these genes are metallothioneins. Metallothioneins are cysteine-rich proteins, so they bind with heavy metals like zinc, cadmium, and copper. So metallothioneins serve as an intracellular zinc reservoir, and also they scavenge free radicals. Many papers have shown that metallothioneins inhibit hepatic carcinogenesis. So, and also in hepatocellular carcinoma, the expression of metallothioneins are decreased. In addition to the cancer tissue, metallothioneins expression decreases in chronic liver disease too. So the hepatic expression of metallothioneins is induced by acute phase response like toxic liver injury or acute viral hepatitis, but in chronic liver diseases, the expression of metallothioneins are decreased. So as you can see here, patients with simple steatosis and non-alcoholic steatohepatitis, these patients also showed that the expression of metallothioneins are decreased in their liver tissue. In addition, as I mentioned just before, metallothioneins serve as an intracellular zinc reservoir, and zinc homeostasis is primarily regulated in the liver because serum zinc is mainly bind to albumin and macroglobulin produced by liver, mainly liver. So if there is chronic liver damage, it results in the impairment of zinc homeostasis and eventually zinc deficiency. Even more, the zinc deficiency evokes a variety of metabolic abnormalities in chronic liver disease. So zinc supplementation has beneficial effects on chronic liver disease. So there is several clinical trials supplement zinc to the patient with liver disease. So in this study, patients with chronic liver diseases were supplemented with zinc. So this zinc group, dot line, shows a lower incidence of HCC, and also when this group measured serum zinc concentration, higher zinc concentration was associated with lower HCC incident rate. So this study shows that the zinc supplementation reduces HCC risk. So in my study, I treated atorvastatin, and then I measured intracellular level of zinc in the cell. As you can see here, atorvastatin treatment increased the expression of this green dot. The green dots indicate intracellular zinc. So this figure shows atorvastatin increased intracellular zinc, and it is probably mediated by inducing metallothioneins by atorvastatin. So this is the summary figure of my study. So statins, including atorvastatin and other statins, commonly inhibit mevalonate pathway. So they can inhibit YAP pathway and AKT pathway, eventually inhibit hepatocarcinogenesis. But this special thing, atorvastatin, has another mechanism of inhibiting hepatocarcinogenesis. So atorvastatin inhibit TRXR alpha mediated AKT activation, and also atorvastatin increased the expression of metallothioneins, and then these metallothioneins inhibit the hepatocarcinogenesis. So my previous lab that I worked as a postdoc is now conducting clinical trial to test the safety and efficacy of atorvastatin on HCC risk. So this clinical trial tries to to try to prove atorvastatin can prevent the progression to HCC from liver fibrosis and cirrhosis. And this is my the final part of my presentation. So this figure shows the importance of non-alcoholic fatty liver disease. So why the reason why NAFLD is important is that in general population, the prevalence of NAFLD is around 25%, and also in South Korea, it is reported that the prevalence of NAFLD is around 30 to 40% in Korea. And this early stage of NAFLD, non-alcoholic fatty liver, progress into non-alcoholic steatohepatitis, and then progress into fibrosis and cirrhosis, and eventually progress into HCC. But the important thing in NAFLD progress is that the NAFLD can progress into HCC directly without fibrosis and cirrhosis. So these characteristics suggest the early management of NAFLD is very important. So, so far, the lifestyle modification is the only way to manage the NAFLD. So, weight loss by Mediterranean diet and regular meal pattern, and avoiding snacking, and the reduction in fructose and glucose intake, and increasing daily physical activity is the only way to manage NAFLD so far. And also, many research groups and companies are trying to develop the pharmacological therapy for NAFLD, modulating these various kind of mechanisms, but there is no approved drug for NAFLD so far. Even more, there are tries using combination therapy for NAFLD that use the multiple drug that can modulate the multiple target. So in this perspective, the herbal medicines are a good example of multi-component and multi-target drug for NAFLD. So recently, many studies were conducted, and this Tansam and Texa has a various kind of mechanism to modulate the pathogenesis of NAFLD. So based on my previous study, I want to develop a herbal medicine preventing NAFLD-related HCC. The herbal medicine contain multiple components, and they can modulate multiple targets, so it can be efficient for liver cancer prevention. So fortunately, I recently got a grant for this study. So I'm gonna screen HCC preventive herbs using literature review and in silico prediction using the PLS system I introduced before. And for the next step, I will find the best combination of HCC preventive herb using AI algorithm optimization. And then I will test the efficacy and mechanism and safety of the drug in these in vitro, in vivo, and ex vivo levels. So if I can develop the novel drug candidate, I would like to progress to clinical trials and hopefully globalization. So thanks for your attention.